Hello, everyone, and welcome to Enabling Oral Delivery of Sensitive Molecules, presented by Capsugel. I'm Morgan Edwards-McGee, Operations Manager of the Life Sciences Group at Fierce Markets, and I'll be moderating the webinar. Our speakers today are Asan Benamer, Senior Director of Pharmaceutical Sciences at Capsugel, and Jim Coward, Senior Director and Global Head of Market Development at Capsugel Dosage Form Solutions. You can read their full bios on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge it. Please also disable your pop-up blocker to participate in the interactive parts of the presentation. We'll follow the presentation with a Q&A session, so please submit your questions during or after the presentations using the Q&A box at the right side of your window. So I think we're ready to begin. So Jim, please take it away.
Thanks, Morgan, and thanks for the introduction, and thank you everyone for attending today as we review Enteric capsule drug delivery technology and its potential applications. Let's first set the stage with what we'll cover today. I will provide some introductory comments on Capsugel Dosage Form Solutions, which is a business unit within Capsugel, before turning the presentation over to Hassan, who will review Enteric capsule drug delivery technology in some detail, inclusive of in vitro and in vivo study results. Hassan has led the development of this new technology and is working closely with lead user applications. And he will also review a case study from one of these lead users that is evaluating the technology for an oral vaccine application. First, a quick overview of who Capsugel Dosage Form Solutions is. The business unit includes in-cap drug delivery and Bend Research, companies that you've probably been involved with in the past. And we follow a very specialized CDMO model for the design, development, and production of innovative dosage forms for our clients. Our proprietary platforms, extensive IP with over 100 patent families, and select internal drug product development differentiates us from some classic CDMO models out there, as does our project philosophy and approach. One, we utilize multidisciplinary product development teams made up of a blend of chemists, biologists, material scientists, chemical mechanical engineers, as well as pharmacists, which work hand in hand with client teams to advance drug compounds. Our teams bring a broad scientific perspective to client problem statements and tend to complement our clients' teams. Additionally, we do not push or promote any individual technology or finished dosage form presentation in our work. We instead choose the optimal technology and approach in meeting a client compound target product profile. We sometimes refer to this approach as agnostic and tend to let the science dictate the enabling technologies chosen to meet the target product profile and client commercial objectives. Additionally, we've pioneered technology selection methodologies based on our collective experience across the legacy companies in advancing more than 1,000 compounds. And we've extensively studied and identified and modeled the key parameters impacting performance, stability, and manufacturability of the full range of oral solid dosage forms. And the predictive models that we've developed typically enable very rapid technology selection and limit the amount of API needed for early formulation work. Additionally, our focus areas are aligned with some of the industry's toughest formulation challenges. Bioavailability enhancement provided by a full range of key enabling technologies, lipid, nanotechnology, solid dispersion approaches, and this capability being complemented by a full range of modified release technologies, of which Enteric capsule drug delivery technology is part of. We tend to view these platforms collectively as often a combination of these enabling technologies are utilized in meeting client problem statements. We've also developed some specialized capabilities in inhalation formulations based on particle engineering from Bend Research for pulmonary and nasal delivery applications, and biotherapeutics with dedicated lab facilities. Additionally, we've invested in development and manufacturing infrastructure for high potency and controlled substances, certainly a key trend as the audience is aware. The next slide gives some detail on our technology platforms and puts into some context how enteric capsule technology blends with our overall capability. We have a full range of bioavailability enhancement technologies and expertise in MR solutions, both in controlled release and delayed release applications, in order to effectively meet client compound target product profiles, inclusive of fluid bed multiparticulates as well as matrix multiparticulates, inclusive of lipid multiparticulate technology and mini-tabs, bi- and tri-layered matrix and proprietary osmotic approaches, gastric resistant capsules, primarily for health and nutritional applications, colonic delivery approaches, leveraging liquid fill hard capsule technology, and fixed dose combinations using both monolithic and multiparticulate approaches, inclusive of capsule and capsule approaches. Now we move into enteric capsule drug delivery technology specifically. First and foremost, it is the world's first enteric dosage form technology providing full enteric protection, not gastric resistance, without the use of coatings. There's been quite a bit of work on this topic in the industry through the years. The combination of polymer science and capsule shell design and manufacturing expertise have come together to yield this breakthrough technology for intrinsic enteric protection and delayed release. The lead users we're working with currently cover several applications, inclusive of the topic of today's presentation, the oral delivery of sensitive molecules, but also rapid prototype development and an in vivo testing tool, and I'll give a brief overview of this application in the summary section. Thirdly, novel enteric dosage forms leveraging the intellectual property associated with this new technology, and ultimately we do see the technology being applied in the future for application in simplifying manufacturing and QBD applications versus enteric-coated dosage forms. So we're excited about the technology, and I'm going to turn it over now to Dr. Benamir, who is joining us today from France. As mentioned earlier, Hassan heads up our pharma sciences team and has led the development of enteric capsule drug development technology over the past several years. Hassan?
Thank you, Jim, for your kind introduction. Good morning, everybody. As mentioned here, it's our pleasure to present you the enteric capsule drug delivery technology. I will start with the definition and specification. On the slide here on the background, there are why enteric properties are needed. In the one hand, there are some specific API attributes that has to go through the stomach and then later on be available in the intestine for absorption. These attributes are, these API could be sensitive to the acid of the stomach, they could be sensitive to the enzyme, they could have some local toxicity, or maybe we're just looking for a fast burst effect absorption in the intestine. Which kind of product can be applied on that? This could be, like we mentioned at the focus of today, for small molecules, biomolecules, peptides and small proteins, or live organisms like bacteria, viruses. Toward that targeted product profile, we developed like mentioned, the first enteric capsule intrinsic, and I will explain what that means as a drug delivery technology. Indeed, on the challenges that we are looking at for solving the problem of oral absorption of peptide, vaccine, and nucleotides, these are divided into two approaches. The first is a safe passage throughout the stomach, indeed, the acidity of the stomach can lead on the degradation. This acidity pH can move from 1 to the highest 3 pH in fastest stage. There's some enzymatic secretion like the pepsin. So unless protected, this acid and the enzyme will degrade the peptide prior to absorption. Our strategy is to say, and there's a lot of literature research, and this has always been like a gold research or approach to find an answer to deliver oral peptide by providing an intrinsic enteric capsule. The first part is answered, the safe passage through the stomach. Then the scientist and the pharmaceutical scientist can focus on the drug delivery and work on the absorption strategy that can occur and will occur in the intestine. So the combination of this unique capsule and the formulated design, the formula for the peptide, will enable to see more product tested and also, and hope, marketed using the oral rule for peptide vaccine and nucleotides. To answer that challenge, the enteric capsule drug delivery, throughout our brainstorming and customer discussion and to define the targeted product profile of that capsule, it came directly that some key attribute has to be defined. First of all, it should be a two-piece intrinsically enteric hard capsule. That means that it should look as a regular capsule, so that there is no new investment, no new equipment to be bought to do or to be able to use that technology, either in preclinical or even in the first clinical and commercial aspect. That's what the first product description need. Secondly, the polymer that should be used should be answering the regulatory status. And we took the decision to use well-accepted cellulosic derivative, and we blend those cellulosic enteric derivative polymer together with no other, this 100% enteric product polymer that is used to make the capsule. Currently, we have available two kinds of size of capsule, the size 0 and size 3, as immediately available and can be used for the first program based on the first lead user program that we had. We are using a white opaque standard capsule. That specific color can be asked upon request. And currently we are using dry ingredient or powder-based formulation only. That means that we don't have enough today compatibility or studies using other form like liquid or other system which could have some compatibility issue that we are currently investigating. As mentioned, the regulatory status is that the polymer used has been on the market more than 20 years, and they comply with the European, Japanese, and USP pharmaceutical and regulatories. The water content of this capsule is less than 7%, and usually can be used for dry or sensitive water product. And the storage is usually the same as used for capsule. The approach is that we recommend to use a more tight packaging to keep, and this is always requested for sensitive molecule. The shelf life and what has been done today, as you can read from the slide, is that we did cover the 6-month accelerated condition. We have currently 12 months at 30, 65, but also at 25. And we have ongoing stability at 5 and minus 20 degrees C, as we've seen that for these vaccine or other live bacteria, the conservation or the storage of the final product is around at cool or refrigerated temperature. So once we have been developing and put together polymer science, engineering, and design to develop the capsule and select the right polymer blend, and when we had the capsule that you have seen on the previous slide, the question came, what should be the best model compound to be able to demonstrate the proof of concept that this is a true enteric capsule. So we went and we decided to use a proton pump inhibitors reference, and we chose the esomeprazole magnesium triedrate that you will see referenced at EMT. The reason why we choose that is that it's really acid label model compounds, and that have a rapid, fast degradation that you can dose, assay by HPLC, but even that you can see quickly through dissolution, disintegration, as your media become yellow to brown very quickly. So that's the selection, the reason why we're using the EMT. And we put it like usual classical making those pellets by layering them on sugar beets. We use those uncoated. Usually the marketed product like the reference, the Nexium, is then coated with an enteric polymer. We did not use that coating. We just filled those pellets, uncoated pellets EMT in the capsule. And we compared to the reference Nexium to see the behavior, how it works, also in vitro and in vivo. The main difference is that the Nexium is using pellets coated in a hard gelatin capsule, which is called a purple pill, and we are using uncoated EMT that we put in an enteric capsule. If the enteric capsule should avoid migration of acidic dissolution medium into the capsule and causing any EMT degradation that we can first see from a discoloration, turning from transparent to a yellow-brown solution. So that's the first test that we did. So we took the enteric capsule drug delivery capsule and we filled them with uncoated pellets, and we run the USP disintegration test monograph that is made for Nexium or any enteric product. So it's first we do the 2 hours agitation in the acidic media, pH 1.2, and as you can see from the picture on your right, the ECDDT didn't no release or open the Nexium or the EMT, while if you compare to what it's called today resistant capsule which are on the market, you see that first of all you have a discoloration of the capsule and degradation, deformation, but also release and the media turn to yellow. Moving to the second step, which is the evaluation of the enteric drug delivery capsule during in vitro dissolution test, we did follow the test of putting the capsule filled 2 hours using the USP2 paddle method. After 2 hours in pH 1.2, the capsule has stayed any evidence return, no release. And then when transferred to the buffer stage mimicking the intestine, pH 6.8, you see that at 30 minutes the capsule has been dissolved. How that look now to the approach of a normal dissolution graph? Here is the reference. So as a start, you see on your left hand the purple pill that we put at T0, which is a gelatin. And of course, the gelatin will quickly dissolve and release the pellets which stay in the bottom of the vessel. And as you can see from the picture, after 2 hours in pH 1.2, we see no gelatin but only the coated, enteric-coated pellets. And then when we transfer them to the buffer stage, you see the rapid release, the complete dissolution of the pellets, and at 30 minutes, 94% release of the EMT. In comparison, we did the study by filling the capsule with the enteric drug delivery capsule with the EMT uncoated pellets. And as you can see, after the 2 hours in pH 1.2, there were no dissolution of capsule, no release, no change of the coloration, and we didn't have zero release of the EMT. Once we move to the buffer stage, we start and we have, after 5 minutes at pH 6.8, you see that the capsule did open and start to release some of the pellets. At 15 minutes, you see that the entire dissolution of the capsule is achieved, which is showing that the capsule did do the job of resisting in the acid and passing the test. Thanks to that good in vitro test, we were directly we have designed the assay in human biostudy, and we did both the scintigraphy and the pharmacokinetic study. The protocol of that study is the following: is that it was randomized, open label, single dose and crossover pharmacokinetic and gamma pharmacoscintigraphy has been conducted using healthy volunteers, 14 people, on the faster stage. The radiolabeled pellets has been added to both the Enteric drug delivery capsule, but also to the reference Nexium in order to detect location of capsule opening in GI tract. The capsule that has been used for the LB, I repeat, are gelatin capsule. So we do then, after the study treatment, we have done some scintigraphy imaging that I will share with you, with the anterior and posterior image acquiring at the dosing, plus regular time points. And we did take blood sampling at pre-dose and regular time point to do a classical pharmacokinetic study. The result of the scintigraphy imaging, as shown inside here, clearly demonstrate that no pellets from the ECDDT capsule released in the stomach. And we had a rapid release from the Enteric drug delivery capsule post-gastric emptying. And of course, the Nexium capsule released in the stomach as designed. As mentioned, these are gelatin capsule, and the Enteric is provided by the coated pellets. So the anterior scintigraphy image indicates that the capsule integrity in the stomach followed by a rapid release in duodenum. As a summary of those effects, we can have the location of the pellets release, and you see that there was no release in the esophagus or stomach from the ECDDT, and majority did release in the small intestine around the duodenum, and one in the ileocecal junction. No release on the remaining stomach after 12 hours, no release in the small intestine after 12 hours. Which is from the opening and gamma scintigraphy. Of course, the majority of the Nexium did release in the stomach or a little bit earlier. So we did achieve in vivo our primary objective to demonstrate the enteric property with the ECDDT capsule that doesn't open in the stomach. From the pharmacokinetic and blood sampling that we did on the two products, we see that first we had, and in blue you have the capsule filled with the EMT uncoated pellets, and in red it is the Nexium filled with coated enteric pellets. We first can see that we have similar absorption with an ECD that have a 30% above absorption, a Cmax which is higher, and a similar peak time or Tmax. We had a similar elimination constant. So despite, and this was a big discussion between monolithic versus enteric coated pellets, we have seen the same approach that we saw in vitro, is that the human PK profile of ECDDT and Nexium showed similar extent of drug absorption and Tmax in a faster state, which was very encouraging on the approach, demonstrating the effect of the capsule doing the job, the enteric drug delivery capsule doing their job, not only in vitro but in vivo. Based on that, we did approach several customers and discussed on how these could be used for their ongoing product and product development. And unfortunately, as you know, we cannot share all this program, but we had the opportunity, and we thank again the customer that gave us the opportunity to work together, not only as a partner, but also to showcase the study so that we can benefit and maybe give you ideas on how this product can be used in your own development. So the product is an oral commercially available vaccine. The product is composition is a gelatin, hard gelatin capsule coated with an enteric cellulosic derivative polymer, contain a blend of lyophilized bacteria, okay, with different blockage and antioxidant and filler and lubricants. Normal powder, classic formulation to be filled in capsule. The coating is done by using organic solvents and did get some, resulting in some bacteria degradation, but also the aqueous coating gives them some challenges for the vaccine viability. So the current product which were commercial were using organic solvents, and the approach to use the enteric drug delivery capsule as a dosage form give the benefit of not using an organic solvent, so there was no residual solvent and had a better, and no coating step, which get a better bacterial viability after the manufacturing, and reduce the step in manufacturing the system. The feasibility consists of, we did evaluate both physical integrity and disintegration, behavior, and capsule filled with the lyophilized formula. After 1 month and stored at -20 degrees C, because those vaccines are going through the cold chain, and it was mandatory and important to show that the capsule did keep that integrity when stored at -20. So we did several time points, and we did the evaluation of the enteric property in acidic classic medium, and then the buffer medium at pH 6.8. We did determine if there is some brittleness tendency, and we did the viability count of the bacteria. The outcome came that the physical compatibility between those lyophilized formula and the enteric drug delivery capsule was demonstrated. We had no increase of brittleness or visual change after the month. We have, of course, this is at the time that we did the feasibility program. We have now ongoing stability, as I will be showing you. The enteric protection of the field content and followed by fast release in neutral media in vitro has been confirmed, and the viability count showed promising results, close to the initial required counts. Here are the more in detail the disintegration with the lyophilized powder in the system, and I want to show here two approaches. And first, that we did it with non-banded or not sealed capsule, and the other time there in second, and with the banded, which have a similar approach. So there is no opening, no sign of cell rupture, and the disintegration medium was totally clear. At pH 6.8, fast opening of the capsule, complete disintegration of the capsule, I think within 7 minutes. The specification is 30 minutes at customer, and just to show you that it was very fast in the approach. As these are lyophilized powder vaccine, there is no dissolution method for this product. So what we did is that we took the capsule size and we made again the dissolution I did share with you with the pellets. This is for size 3, and we demonstrated and confirmed again that there was no solubilization of the capsule. The dissolution media was clear in acidic media. There was intact EMT release, no degradation, and the total release was we cannot detect, is below our limit of detection. So in the pH 6.8, in the buffer stage, mimicking the stomach, we had again a rapid release reaching to 98% after 30 minutes. Based with those very encouraging, the customer has decided to proceed to the larger manufacturing and to produce batches under controlled manufacturing condition for full formal stability program. So they have performed 3 stability batch under controlled manufacturing condition by filling the Enteric drug delivery capsule with the current market formula at their site, and they did run 100,000 capsules to perform that. So the flow diagram to do that, so the marketed lymphocyte powder blend has been prepared, filled using a high-speed filling IMA machine. Intermediate packaging has been done, sealed in alu bag and then put in freezing. Then unfreeze product later on has been done for packaging in blister, and then put in stability program at 5 degrees C and 25 degrees C, 60% relative humidity. The key outcome is that the capsule was sorted and printed on their adequate machinability was confirmed in-house. The first stability batch was successfully manufactured by the customer on their IMA-matic machine, like I mentioned. The capsule was successfully fed into the machine, rectified, open, filled with the formula and closed, and then rejected at an output of 70,000 capsules per hour without any machine stop. And the first data of stability point show a promising initial viability data. This is again encouraging and showing that not only the product is answering from a lab scale, but can run on existing machine at a decent speed when having the right adjustment made on the machine. Having said that and presented the technical part, I will pass over to Jim to discuss about the summary and the point and next step forward for this technology. Jim?
Thanks, Hassan. We thought it'd be worth a quick overview of another potential application, which is utilizing enteric capsule drug delivery technology for a product development tool with speed to clinic implications. So just a quick overview here. Capsugel commissioned an assessment by H2 Pharma Consulting in the UK, which is Dr. Heap Houghton, that many of you may know, to assess and confirm the internal analysis that we've done on the relative speed to clinic savings and development risk mitigation in eliminating the need for enteric coating of drug products containing acid labile or stomach irritating APIs and or drug products that required specific targeted release in the upper GI tract. Heap has significant experience with Pfizer, Focus, and other consulting engagements, and has deep expertise in formulation development and modified release dosage forms. So first, some background, of course, in developing a product requiring enteric protection or delayed release, there are several challenges. As the audience likely appreciates, enteric protection has historically been exclusively provided by enteric coatings, solvent or aqueous-based, with relatively high weight gains versus standard or immediate release film coating, requiring a minimum of 10% weight gain and as high as 20% or more for multi-particulate applications. Oftentimes, multiple coating layers can be required, for example, seal coat applications, and sometimes curing requirements for aqueous-based systems. The formulation, process development, scale-up, and validation steps in preclinical development carry the highest development risk and limit the speed of development, ultimately progression to clinic. These phases, therefore, offer the greatest scope for optimization and de-risking the formulation development with a technology that can provide intrinsic enteric performance. So that's the premise that we're operating under. Some detail on that, some background. In the formulation and process development phase for enteric products, there exists the potential of significant time to select a formulation. There's likely a need for multiple prototypes to achieve desired in vivo performance. There exists the risk of functional performance changing on scale-up, given the intrinsic link between formulation and process. And scale-up and validation, there's likely the requirement for multiple coating step at development. need for comprehensive design of experiment for process optimization, process validation for the coding steps, and scale-up studies required prior to bioequivalency evaluation. And finally, in clinical and bioequivalency testing, iterative changes to formulation to achieve bioequivalency may be required, resulting in significant rework of process steps, scale-up, and potentially process validation. So listed on the slide here are the potential advantages associated with intrinsic enteric functionality afforded by enteric capsule drug delivery technology in terms of reducing the complexity of enteric dosage form design through clinical and bioequivalency testing, and allowing ultimately rapid prototype development and in vivo evaluations. So net for new chemical entity development, H2 Pharma has estimated 9 to 14 months net savings associated with formulation screening, process development, process scale-up and optimization, process validation and PAT/process control steps afforded by the intrinsic enteric technology. This net savings in development time and de-risking several areas of the development cycle may be especially critical for 505(b)(2) applications, given the acute time and cost concerns of this particular approval pathway. Now let me speak a little bit about the lead user program that we've been under. This was expanded last fall. We had a press release on that. We've evaluated, gone through a number of lead user programs at this stage. We're in the final stages of the technology's development. The term lead user really indicates that our work with clients entails their providing feedback on the technology's performance in their applications, and we're working hand in hand with clients on their projects. Our teams, our product development teams, are quite active with enteric capsule technology projects, working with virtual to large pharma clients across a range of APIs, applications, some of which we talked about today, and finished products. We see interest in application in both small and large molecules, enteric protection, targeted release, and as a preclinical product development tool, as I just walked through. So in summary, before we take some questions from the audience, enteric capsule drug delivery technology is novel. It's proprietary breakthrough technology providing enteric protection and or targeted release. As Hassan has walked through, the enteric performance has been confirmed in vitro and in vivo and continues to be validated with lead user projects. We see a varied application for the technology in our product development with clients, inclusive of being truly enabling for the oral delivery of sensitive molecules. Preclinical development tool, we're seeing application as I've mentioned, and enteric product and performance differentiation, potentially leveraging this technology's intellectual property. So the lead user program is well underway. The technology will be featured at upcoming events at Controlled Release Society. We'll be featuring the technology. We'll have a poster there. At CPHI, it will be in the Innovation Gallery. At AAPS, we have posters on the technology and a seminar as well. So I look forward to seeing you at some of those events. If you'd like further information, certainly contact us at the email given here, dfsinquiry@capsugel.com, and we'll have one of our Capsugel Dosage Form Solutions business development representatives get in contact you in the near future. So this concludes the presentation component of the webinar. Thanks very much for your attendance and attention, and I'll turn it back over to Morgan, and Hassan and I will be happy to take any questions from the listeners. Thank you very much.
Great. Thanks so much, Jim and Hassan, for those great presentations. We can move on to the Q&A portion now. We have a lot of great questions, and we'll try to get to as many as possible. So I'll jump right in here. First question, if the capsule contents aren't coated, shouldn't the risk of dose dumping be huge? And how do you ensure such a risk is mitigated if the capsule were to rupture prematurely?
Yeah, Jim, I'm taking that question. So indeed, that's a key question. That's why through our process and in-process quality control and using the Six Sigma approach and quality by design, we make sure that those capsules, or the thickness like you do for coating, is ensured in all the aspects. So we don't see, and we didn't see now all the tests performed to date with our product during development, but also at customer and in our in vivo study, we didn't see any opening or dumping or puncture that get it de-release premature. So if they are handled properly, like we do for any coating, or like I said, the vaccine product that are on the market that we use as the key study. I just put the slide back. This is a coated product. So the difference here is that instead of having a gelatin coated product, we have the capsule per se intrinsically is made of 100% enteric polymer.
Great, thank you. Moving right on to the next question. Do you have experience with protein delivery with or without permeation enhancers or protease inhibitors?
Yeah, looks like it's again for me, Jim. Sorry, I'm taking that question to get you that. Yes, I think the work that we are doing is that there are protein or small peptide, and we just want to make sure that this is the approach in which we're working in. And the experience we have is using a lipid-based system, which in some cases could be close to permeation enhancer, or could be only taking the advantage of the absorption promotion of the lipid throughout the natural part of absorption that could be used to enhance the protein delivery. So that's the experience we have currently in conjunction with other formulations that are using permeation enhancer. So both can be done. The blend and the quantity, that's the challenge that has been done as a drug delivery. By using the enteric capsule, like I mentioned in the presentation, I think the advantage is that we can focus and to optimize that formulation delivery to make sure that we have that in vivo test quickly, and we can turn different formulation to select the lead formulation one. Currently, what people are doing, this is the rate limiting factor, is that they have to tackle the both challenge with the oral of peptides, is the safe passage through the stomach and the effective absorption. Here the advantage is that we can focus and work only on the second step, which is increasing the rate of success of bringing product to the next phase or to animal study or human clinical trials. So that's my answer.
Okay, thank you so much. Okay, moving right along to the next question. Does ECDDT resist deformation and leakage at the capsule joint under acidic conditions, and for how long?
Sorry, I was on mute. That's a question for me, I guess again. So today what we have been showing is that we have demonstrated or reproduced several batches production and capsule through development and manufacturing, that they all resist easily 2 hours without disruption, without opening. And we did some more extensive tests in acidic pH up to 22 hours, 24 hours without any opening of or disruptor of the capsule in the acid condition. The acid condition test was from 1.2 to 4.0, and the capsule did resist that, like I said, from an approach of 2 hours minimum up to 24 hours.
Thank you. Okay, this one might be for you as well, I'm not sure. What do you mean by in the final stages of product development? Are these capsules available off the shelf?
I can take that one. The capsules are not available off the shelf per se. This is a drug delivery technology, so we have development agreements with clients, including this lead user program, with which joint evaluation of the capsule shells are performed.
Okay, thank you so much. Okay, moving right along here. What is the highest pH that you've evaluated for dissolution? The technology's formulation is sometimes patients have an elevated stomach pH.
Yeah, that's a question that I'm taking, Jim. Here we did tested, so like I mentioned, pH 1.2, okay, pH 3, 4, 4.5, 5.0, and 5.5. So on all the pH below the pKa, so up to the pH 4, the capsule did resist. And from the literature and the discussion we had with experts in the in vivo in human pH measurement, we know that the pH can increase to up maximum pH 3, so we test to pH 4, and usually people have to test and to see that the capsule does resist at that pH. So we did show and demonstrate that the capsule does resist in pH 4 with 2 hours with no release, and at pH 5 or close to the pKa, which is a 5.5, we start to get the opening and release of the capsule, which is in line of the design of that product. The objective is to get it released in the duodenum quickly, where we know that pH is around 5.5, and that's key.
Okay, thank you. Next question, and I think you might have alluded to this before, but just to be sure, do you need a sealing step for the capsule after encapsulation to prevent gastric juice from seeping into the capsule through the junction of the two-piece capsule there?
That's a very good question. I can take this again. So as I mentioned during all, or maybe I forgot, but I will reiterate, during all our in vitro tests, so all the data that we shared with you, the dissolution curve, the in vitro, and even the disintegration, were done without, and I think it's important to show this slide, have been done without bending or sealing. So as you can see that the capsule did cut them to show the inside, the capsule did not show any evidence of water or acidic getting into the capsule or make any degradation of the system. On the other hand, our clinical or human biostudy has been done and made with a sealed, banded, more specifically banded capsule. The main reason, because it was our first study, we wanted to make sure that we didn't see any, because the hydrodynamic condition in vitro and in vivo are quite different, we wanted to make sure that there isn't any opening, and they did pass the test. So currently our advice is the following: unless we demonstrate it in the future clinical study, is that for the in vitro test, dissolution and disintegration, we use unbanded capsule or unsealed. In vivo, we are using band one. We recommend to use the banding one. There are currently some customers that decided to go in clinical because the data they've seen and the result they've seen in vitro and knowing their product, they're quite confident that those products will not be sealed or banded. So we like Jim said, this is an ongoing project. We'll see, we present CRS, IPS, so you will see some of those results in the coming symposia or press release that we'll be making while we're developing the product.
Okay, thank you. Next question, was the banding done with enteric capsule material as well that would survive the acid phase?
Yes, it was.
Okay. And can I further coat the capsule to create additional delay of drug release for an extra 4 hours so that I could also use it for colonic delivery of the API?
Jim, you want me to take that, or because there's two angles to answer. We have technology that are which are more in our portfolio, like Jim has said, for colonic drug delivery. We are not intending today to do or recommend to do any coating on those capsules, because usually what we see on doing the capsule, we are introducing that the drawback that I was explaining on the adhesion of that new film on the capsule, the integrity, to keep the capsule integrity using water on that. So the approach is that, and this leads to a new discussion, will be a colonic delivery capsule will be available. We are working hard on it, so stay tuned. If there is a lot of requests, we'll be able to come in the coming years with the colonic drug delivery. But we have alternative technology to go to colonic delivery. Maybe Jim, you want to comment on that?
Yeah, there's as Hassan says, there's some development work ongoing, but we do have approaches in our kit today for colonic delivery. Some of them utilizing liquid fill hard capsule technology. We also have cap and cap technology that can be utilized. So some of these questions, of course, we'll have some specific follow-up with the individuals asking, and can get into some discussion about what exactly is needed for your particular projects, and there's a number of tools in the kit with which to achieve those.
Wonderful. Thank you both. Next question. What is the range or limitations for enteric capsule sizes and colors?
So currently the limitation is time to produce them, to make them happen at the quantity. So basically when we are able to do a size 0 on our industrial machine, commercial machine, there's no limitation on size of color. Technically, let's say, is more that we are talking about 2 to 3 months delivery time to get this color and this capsule available. So it's mainly time than technical issue.
Okay, wonderful. Next question, how do I get samples?
We discussed that one earlier, but it's
my apologies.
These aren't samples supplied in the manner that we do with the hard capsule portfolio. This is a drug delivery technology, so it's a discussion on an individual project basis and the tailored to the client need, and our product teams working hand in hand with the client product teams on the evaluation, and of course samples are provided within that context for a joint evaluation.
Got it, thanks. You were talking about pH levels earlier, but the next question also discusses that. I just want to be sure that this was answered. How does Enteric perform at an intermediate pH, say 4 to 5?
Yeah, I think we had I will reinforce. Apparently, it's important. So at pH 4.0, there is no capsule deformation, no release up to we tested, like I mentioned, 2 hours up to 24 hours. So there is in that 4.0 no release on that. At pH 5, in which we are close to we are at 0.5 units lower than the pKa. So some of the functional group have been ionized, so we have a release. So we had about 5% released after 2 hours, and almost majority after 6-7 hours. The capsule was totally dissolved.
Okay, thank you very much. Okay, moving along. This will be our final question, I think. We have a lot of great questions today. Couldn't get to them all, but we'll be getting back to everyone who submitted questions personally via email afterward. So moving right along here, are there any API or excipient restrictions?
Yeah, that's for me. So currently we didn't see any API restrictions, but that's why we are always careful, and at Capsugel for all the capsule program we're running a capsule compatibility. What we didn't, for the time being, we do not promote filling using a liquid excipient or surfactant or highly hydrophilic excipient due to that compatibility constraint that we should be performing before we allow that. And currently, the targeted product, like we said, vaccine powder, these products are in the powder form, and that's the one that we encourage to do first, to save time and money there to do the program and confirm the proof of concept. So I repeat, from the API, we didn't see any, for the time being, constraint, but we are running what we call a compatibility program. That's the minimum to be performed, and we do not recommend using lipid or liquid excipients.
Great, thank you. I'd like to thank our audience for submitting so many great questions and for attending the webinar today. I'd also like to thank our speakers for participating and Capsugel for presenting today's webinar. The webinar has been recorded, so you'll be able to access the recording within 24 hours on the same page that you used to register for the event. So thank you again, everyone, for joining, and we look forward to seeing you at future events.